DNA cloning involves 3 steps. First, DNA is isolated from an organism and cut into pieces using restriction enzymes. Second, each piece is inserted into a cloning vector capable of replication in a host organism. And third, the cloning vector containing the DNA fragment is introduced into a host, such as a bacterium or a yeast, animal, or plant cell. Let's look at each step in the cloning process. Restriction enzymes recognize a specific base pair sequence of DNA, called a restriction site, and cut the DNA within that sequence. Many restriction sites are symmetrical about their midpoints. That is, the base sequence from 5' to 3' on one strand is the same as 5' to 3' on the complementary DNA strand. Many enzymes make staggered cuts in the DNA, producing fragments with overhanging or sticky ends. For example, EcoR1 leaves 5' overhanging ends, whereas PST1 leaves 3' ends. Other enzymes, such as SMAL1, cut midway within the restriction site and produce DNA fragments with blunt ends. In a cloning experiment, the first step is to cut the DNA into pieces with a restriction enzyme. This produces a population of DNA fragments in different sizes, but with one thing in common. Each fragment has the same sticky ends. The second step is to prepare cloning vectors to receive the DNA fragments. Bacterial plasmid vectors are most often used in cloning. Plasmids are circular, double-stranded DNA molecules that are independent of the bacterial chromosome. DNA fragments up to 10,000 base pairs long can be successfully cloned in plasmids. Plasmids have 3 important features. First, an origin of replication required to replicate in bacteria. Second, a selectable marker that enables scientists to distinguish bacteria harboring the plasmid from bacteria that lack the plasmid. The usual selectable marker used in plasmids is a gene for resistance to an antibiotic, such as ampicillin. The third feature plasmids must have is a multiple cloning site. This is a cluster of unique restriction enzyme cleavage sites for a number of enzymes. One plasmid vector often used for cloning is PUC19. It has an origin of replication, an ampicillin-resistant selectable marker, and a multiple cloning site for more than a dozen restriction enzymes. The multiple cloning site is located in a region of the E. coli beta-galactosidase, LAC-Z+ gene. The plasmid was engineered so that when a piece of DNA is cloned in the plasmid, functional beta-galactosidase cannot be produced in E. coli. We will shortly see how this LAC-Z+ gene is valuable in cloning selection. We cleave plasmid PUC19 with the same restriction enzyme used to cut the DNA. The DNA fragments are mixed with the vector pieces in the presence of DNA ligase. Because they have identical sticky ends, the 2 DNAs combine. At the same time, some of the PUC19 molecules recircularize without including a piece of DNA. The plasmids are introduced into E. coli by transformation. The transformed bacterial cells are grown on nutrient medium containing ampicillin and the colorless chemical X-gal. Only cells containing PUC19 can grow on ampicillin because the vector contains an ampicillin resistance gene. X-gal is used to distinguish the bacteria with recombinant plasmids or just PUC19 alone. The cells with PUC19 and no DNA insert have a functional LAC-Z+ gene and produce beta-galactosidase. Beta-galactosidase converts the chemical X-gal to a blue color so the bacterial colonies become blue. In cells with the recombinant plasmids, DNA is inserted into PUC19 and disrupts the LAC-Z+ gene, so no functional beta-galactosidase is produced. X-gal remains unchanged and the bacterial colony appears white. This X-gal test is a simple way to distinguish bacterial cells carrying recombinant plasmids from those that do not. It helps us identify the desired recombinant DNA clones.